Hi, hello everyone. I'm Kate. Kate Song. I teach at Castaic High School currently, but I was at Santa Monica High School for 19 years. So I'm going on, I think, 23 years now as a teacher. 21 of those years were teaching AP Bio, where I started implementing this biotech lab. So I've been doing the biotech experience for about 20 years or so, a while. So Karen asked me to come and kind of talk about my experience as a veteran teacher implementing this and how I use it specifically for my AP Bio students. So that's the audience. And here's some pictures that I gathered a couple of years ago. So these are the bright, happy faces of students that successfully completed the series. I do the full version of 1 through 5, labs 1 through 5, not the abridged version. So we actually cut the recombinants, we recombine them, and then we transform them into bacteria. And then this is the day right here where they get to check whether or not they successfully transformed the bacteria into red colonies. And that was one of the teams that succeeded, so bright, happy smiles. This is kind of what we see in the lab, with the jelly electrophoresis and the kids, being really careful and trying to get the tiny little volume into a little well. And I tell them, like, I try to make it fun, and I say, don't mess up. If you mess up, you're going to screw it all up for your group. And so I kind of give them that added pressure, so their hands are nervous, and it's all good fun. So yeah, that's a big highlight. And then this is one of our gels. I put 2 groups on one gel, so less gel prep. And then the picture that we take, and then I upload it to, we use Canvas, but you can upload it to whatever, Google Classroom, and then students can analyze it. And I don't know if you can see, this is one of our successful plates. You can see the red dots, lots of red dots. So that was a really great plate. And one of my students came up with a meme at the end of the year, because I like to call the red colonies our babies, so that they have this connection to their product. And so she came up with this red baby meme, and I have it on my wall. So, kids are excited that they made red babies, and I keep reminding them, just remember, bacteria are asexual. They're not really babies, but you know what I mean? So I always refer to our red baby lab, and they know exactly, what I'm referring to. This picture especially near and dear to my heart, because this guy right here is my very own son. So when I started this program, I had just gotten married, and didn't have him until 6 years later. So 20 plus years later, this guy took my AP Bio class, and he was one of the successful teams that got red colonies. I'm like, yay. Yeah. All right. So, have you implemented the lab yet, or is this okay? You're just starting to. Okay. So when I first started implementing the lab, it was a really daunting experience. It's just a big challenge. There's a lot of setup. You're probably going through the motion of actually being the student and going through the lab, so you understand there's a lot of moving parts. One of my coworkers was already doing the lab, so I was lucky. I could watch him do it, observe, and I took notes, and I took the training. But even with that, the first time I was, trying to do it on my own, it was scary. There's a lot of different things going on. Now that I'm at year 20, it's still a lot of different, things that I have to do, but I understand what needs to be done. And so I think having veteran teachers kind of go through the process, I hope gives you a little bit, ease of mind that it can be done. And I've done it in a 55-minute period and in a block period. So I'm going to talk about both experiences, and I've actually prepared pacing guides for either one, so you can see how I run it with a 55 versus a 90-minute block. All right, first of all, when do you order the kits? So I follow the AP curriculum and the sequence. So let me scroll down. This is my agenda, weekly agenda. Sorry, I can't seem to, okay, there we go. Okay, so this is toward the end of first semester, and you can see, like by week 16, I'm already covering DNA, gene expression, and then after my DNA unit, we cover biotechnology, I think at the beginning of second semester. So we talk about gene regulation, and then there's my biotech unit, which starts week 2, second semester. So the reason this is important is because you want to cover the content and then do the lab afterwards. And I usually order the kit in February. So in January, I cover biotech, I test on biotech, and then I think we do the lab. And it's okay to have a gap. Oh, and then I also do a Socratic seminar on CRISPR-Cas9 and bioethics of using gene editing technology. And then, okay, this is where we start our, lab. And you can see that in my agenda, I have exactly what lab we're doing, what day. And then I also have the assignments that I assign on a daily basis. So you can see I start off with, I start off with 1.2, which is basically the practice of micropipetting. So I spend a full period or a full block period just practicing using the micropipetter so everybody's comfortable. And then we also digest the plasmid that same day in a block period. So we practice for about 45 minutes, and then 30 minutes they're digesting the plasmid. And then I also have exit slips, and they just answer the question on a scratch piece of paper before they leave. Well, actually not before they leave. This is, I think they do it at home. And they turn it in for 1 point. We're standard space grading, so we grade out of 5 points in AP curriculum. So I make each exit slip worth 1 point. So I think at the end of the lab, I collect 5 exit slips. So it's worth 5 points. I also leave the lab manual link right here along with the lab PowerPoint. Actually, I give before I even start this, let me show you what else I do. Okay, so how many of you are doing block periods? How many are doing like 60-minute? Okay, so in a block period, it takes 1 to 2 weeks to complete the whole thing. And then in a regular period, again, 1 to 2 weeks. Oh, I thought I had the, let me see.
You can go up to 3 weeks though.
Yes. I need to pace myself though, because my end goal is to get them ready for the AP exam. So I'm very limited on time. So my goal is to finish everything before the AP exam. So I need to squeeze, just even 2 weeks is a lot for the lab. The other thing you could do is you could do the biotech lab after AP exams. That way you get, enough time to finish everything in a relaxed manner. You can also do that. Sorry, I was trying to find, I can't find it right now. I was trying to find the questions that I have for, maybe it'll come up later. Okay, so what do I do in preparation for the actual labs? Do you guys have to prep your own labs, or do you have a lab tech? Santa Monica High School had a lab tech. So when I was at Santa Monica, I would just give him the kit notebook, and he would aliquot everything for me, and then also prepare the gels for me. So I was spoiled for 19 years. And then when I went to Castaic High School, I'm like, what? High schools don't get a lab tech? I didn't know that was a luxury. So starting 3 years ago, I had to start making all of my own gels, which was not a bad thing. It's actually not difficult, but definitely before I start the week doing the biotech lab, I go in the weekend and I prep the gels. So that takes a good hour or so. But the kid notebook, I mean, that's the Bible for you. You just have to, it's so well organized with pictures. I'm sure you guys will get to see it, but you know how like we tell our students read the directions, and when they don't, we're just like, it's right there, just telling you exactly what to do, right? Same with teachers. Sometimes you want to skip the directions because you've been in the training. I remember how to do it, but read the directions. Like if you go by the directions step by step, the kid notebook does everything. Like it outlines like every single detail for us, and I so appreciate like the pictures and everything. So let me show you just like where I go, and I'm having trouble with the scrolling. Okay, I'm not used to, okay, I think I got it. So I just want to show you, yeah, here. This is the gel prep pages. I just go through the pages and I read every single step when they tell you to mix things and prepare solutions. I do the math, just like we did in like college, right? I write it all down, and then I prepare the solutions. You have to put the stuff in the microwave, so make sure you have access to a microwave. You also need access to a fridge and a freezer, so that's really important. I know not every classroom comes with one, so make sure you have a working freezer. And an ice machine would really be great too. Like my campus is a brand new campus. They didn't have an ice machine the first year, so I had to go to the grocery store and get a bag of ice and crush it myself. So just know that those are things that you need to prepare yourself. So this is what I mean. So pictures, steps, and it tells you exactly, what equipment you need. So biggest advice is read the kit notebook and just follow it step by step, okay? And if you have any questions, your site coordinator, Karen, is great. Like I text her and immediately she'll like tell me like what to do. So thank you for that. That's been like every year, she's been really good about responding. So that's the first thing, is prepare the gels. You need to figure out how many gels you need, because the first lab is practicing loading of the gel, right, with 3 different solutions. And then the other one is when you're, lab 2 or lab 3, when you're verifying, or lab 4 when you're verifying restriction. So you need another gel for that. So you need 2 gels per group for the entire series. So I prepare them all at once, and you can refrigerate them. So that first day of preparing gels takes a while. Calculate how many gels you need. Remember, you can share gels between groups, so that makes it, a little bit less work. I prepare a couple extras just in case they break. Yeah, so that's, I think, the most work-intensive day for a teacher in terms of prep. And then the other thing that I do, if you go back to each day, they tell you what to put on the lab station for the students. So for example, lab one, they tell you this is what you need. So I actually have really cheap cafeteria trays that I purchased on Amazon. And I just put all of the stuff on the cafeteria trays. And that makes it really easy for me to just kind of move when I'm prepping. Yeah, so these pictures are great. Instead of a beaker for a waste basket, I just use one of those red plastic cups too, and I just label it waste. Yeah, so follow the directions. That's like my biggest advice. And then, okay, so how do we know that the students are learning besides the exit slips? I used to do this pretty intensive packet where I have students, and I actually recommend this if you're doing this for the first time. I recommend assigning pre-lab questions where they're reading the pages in the lab manual ahead of time, and they kind of get familiar with the whole concept of genetic engineering. And then also, before each lab, they kind of are starting to think about the concepts. And this is the standard that I have up here. Explain the use of genetic engineering techniques in analyzing or manipulating DNA. So everything goes back to that standard. So when we're reviewing, I'm like, okay, this goes back to, you know, the whole point is that we're manipulating, cutting DNA and recombining for a useful purpose. So I'm going to have this available for you all so you can use it. And then my pacing guide actually tells you exactly when to assign what part of this packet. So actually my agenda should. Okay. Oh, this is a different pacing guide. I have another pacing guide that actually has the pages of the packet that I assign before each lab. I don't think I included it here. And then I also have the answer key for the student question. Now, as a teacher, I can't grade all the questions. So what I tell the students is I randomly pick questions that I grade. So the ones that I highlight are the ones that I think are more they're more standards based. So these are the ones that I grade. Kind of saves you time that way. So they hand in the packet when it's due, and then you just grade the ones that are highlighted. And then I also have them like take pictures of, or I post pictures of the gel so that they can, even if you don't have time to run the gel in class, you have the pictures for them to analyze on your Google Classroom. Okay, so if you're doing the 50-minute class, there's ways that you can save time. So you start running the gel and you say, okay, I'm going to stop running the gel after 20 minutes. I'm going to post the pictures and you guys can answer the questions at home. Okay, the aliquoting guide. Okay, so this is the other thing that you need to do before each lab. So the gel prep is like one separate day that I would recommend you do in preparation for the entire series. And then you have daily prep that you need to do. And for that, they have this aliquoting guide. So this is the tubes that you're going to place at each station. So each group is going to get these tubes. So you're going to put 1, 2, 3, 4, 5 tubes at each station. And in order to do that, you need to aliquot 10 microliters of the 1.5 that are given to you. And then they actually only use 8. So there's a little bit of extra in each tube. I do tell the students the reason for the small volumes. Like I ask them, like, why are we using such small volumes? And you know, they try to guess. And then in the end, I tell them, this costs money, right? So everything costs money. So, you know, when we're using these small solutions, you know, that's one of the big reasons. So, you know, like when you're discussing like the volumes and everything, always tie it to like some kind of purpose, right? Oh, and talking about money, I tell the kids before we start that the equipment that they use is really expensive, you know, and high tech, and most college students have access to this, but high school students generally do not. So I let them know how privileged they are in using the equipment, and they understand that they're special for getting to use them, and I say it's really, really important that we treat everything, you know, like with respect. No, like if there's any kind of horseplay at any moment, I let them know you're sitting down, you get a 0 for the lab. So, you know, just let them know that the equipment is expensive. And also when they're micropipetting, a lot of times like they'll just like leave it somewhere, or they like even put it upside down. And I say, no, don't do that, because then the fluid's gonna go into the, you know, the micropipetter, and it could screw it up. So I'm like, just, you know, be really mindful that what you do and how you take care of the equipment, you know, is gonna affect the quality for, because they're gonna be used in other schools. So my students understand, like, this is a pretty special lab. I hype it up like pretty much all year. Like starting day one, I tell them, you guys are so lucky we're gonna do this really fun, cool lab, you know, and we're gonna learn about really cool things, DNA and how to manipulate. Like I actually talk about it like pretty much all first semester, so they have something to look forward to. And it actually is probably one of those things that they will remember. They won't remember anything else in AB Bio, but they'll remember, oh, I remember that cool lab we did, you know, with the red babies, right? So yeah, it's definitely a highlight. And then offering them that extra credit if they are successful with the transformation, that's an added incentive for them to really kind of keep up with the reading. And also, I tell them, like, read the directions before you come to class the next day, because if you mess up a single step, you know, that's going to affect your success at the end. So everybody's like, oh, okay, we better know what to do, especially if you're doing the 50-minute classes. Then you're really, like, short on time, so, you know, they have to kind of know. At least for the 50-minute classes, I definitely would assign the pre-reading and the pre-labs so that they're really familiar with all the parts. Okay, I'm talking kind of all over the place. I hope I'm making sense. Okay, let me go back to my agenda so you can see exactly what we do per day. Okay, so once I have the gels all set up, like I said, I run just the pipetting practice, so that would be one day. And then if you're running a shorter period, I would do the digesting of the plasmid a separate day, so that would be day 2. Then day 3 would be building the recombinant. And day 4 would be the verification, so this is when you're running the gels. Okay, so here's a way I save time. So you're supposed to do the ligation first and then the verification. Because I do them both on one day, we don't verify ligation, we just verify restriction. We just verify that they actually got cut. And that kind of saves me a day by doing it all together. If you are doing the shorter period, I would do the ligation first and then the verification. That's how I did it before, but because of the block and I want to get everything done in a short amount of time, I do the 2 labs together. And then lab 5 is the most complex lab in terms of the steps that the students need to do. So make sure that the students read up on the procedures for lab 5, because transforming the bacteria has a lot of steps, and the temperature difference, the heat shocking part, I think is the most crucial part. So make sure that your water bath is set up to exactly, what is it, 37? 42 degrees Celsius, and then you have the ice ready to go. That's really important. And I explain like why the heat shock is so important when I go over the PowerPoint that's on the Amgen website. So they understand that it's really important to keep them cold. Don't touch the bottom of, you know, the tube because of your, you know, your body heat. Bring the ice bath with you to the water bath, and then dunk it in the water bath and immediately put it back in the ice bath. And then incubator. That's one thing that my school has. It has a little oven where we put the plates. I don't know, do you guys provide those ovens?
We do have.
Okay, so if you don't have an incubator, make sure you request one, because I know not all school sites have one. So overnight we place the Petri dishes in an incubator, and then the last day for us is basically just checking if there was successful transformation. That's the exciting day. Like, that's when And I do the lab with them too, that's another thing. I don't recommend that you do the lab with them the first time around, just because you'll be so busy kind of like monitoring everybody and making sure, you know, everything is there. But now I do the lab with them so that if I don't get any transformants, then I am, oh, okay, then maybe like let's try to figure out what happened here. And that's another thing. If you don't get successful transformation, I know it's disappointing, but I always tell the students this is a learning experience, learning opportunity for us. Sometimes in science we don't get what we expect, but let's think about what happened, you know, and then I have them list like potential things that could have gone wrong, and that makes them think, I think, even more deeply into the process. Any questions? I know that was a lot, went over. I think staying organized is key. Like you can tell, like just from the agenda, I'm very organized and I'm a control freak. So if I don't have everything outlined, I panic. Yes.
You said we're going to have access to this.
Yes. So I'm also going to find the actual agenda that has the pages of the lab manual that I assigned for reading and the pages in the assignment that I give out. I thought it was in here, but I think I might have forgotten to put the link in. I highly recommend, if you have a veteran teacher that you know runs this program, like connect with them, and that helped me out a lot, just having someone on site actually that did the series. Yeah, so definitely having gone through it, I would, yeah. It's so much easier to explain it to them. I actually have a colleague at Santa Monica High School who took on the AP sections that I used to teach, and she's new to the Amgen program. So the first year she was always like asking me questions. And now she loves it. She's a pro after the first year. Yeah. And even though it is a lot of work, I promise you it is so worth it for the students. Like when you see them like operating, you know, the pipetters and then the gel, and they get to see the digest and then the transformation day, it's super exciting. So yeah, I think it's definitely worth the time. It is a lot of time, but you know, anytime you can apply what you're teaching in a way that like, ooh, this is cool, you know, we should do it.
Kate's gonna go to lunch with us, so you can ask her questions then as well. One thing, she has had things go wrong before, and I've worked with her to troubleshoot.
Yes.
So she's very good about keeping results. If something goes wrong, I need results to see, so I can work out what happened for you. Sometimes they've been my fault, so we do try and do quality control testing on all the reagents we send out prior to giving them to you. Sometimes in that stuff, the springtime gets really busy, so sometimes we'll send plates out that I may not keep, because there's like 10 teachers. But yeah, so we want to work through. That's where I told you, you're not done training tomorrow. We'll train you every time you do the labs, and that's where it comes in. And she is correct. When you make a mistake and you try and figure out what happened, you learn the labs much better.
Another thing that's great about this program is not only does it end with the lab, I get opportunities for my kids all the time. Karen sends me scholarship information, workshops for students. This one time we were able to, I don't know if you still have this program where we can invite guest speakers from Amgen. So I connected with a researcher, and he came to my class like every year for like 5-6 years, talking about doing research at Amgen. 2 of my students participated in the entrepreneurship workshop that you publicized, and one of them won first place out of like 70 plus teams, and she was so excited, and she said it was fantastic learning for her. She never even thought of going and like thinking about business and biology together. So there's constant opportunities through this program that you can share with your students. Yeah, so anytime Karen asks, like, can you help me out? And I'm like, okay, I'm there, because I believe in this program. It's, you know, it's been such a benefit for my students and my own kid. He's actually now at UCLA doing biology. He just started freshman year, so I'm excited. Thank you. Oh, and it's 12:30. Sorry, I kind of was all over the place, but I will definitely share this out with you and find the other pacing guide for the regular and the block. Okay?
We're ready for the kids downstairs. You can just take the PPE off and clean your hands.